Welcome to the Microscopists, a bite-sized bio podcast, hosted by Peter O'Toole, sponsored by Zeiss Microscopy. Today on the Microscopists.
Hi, this week on the Microscopists, I'm chatting to Jennifer Waters of the Nikon Imaging Center at the Harvard Medical School. Tune in, watch, listen, and find out what Jennifer's favourite type of microscope is.
I feel like wide field is overlooked too much.
The satisfaction of working in a shared resource core facility.
And we can help them. You know, we can turn that around and put them on the right microscope and show them how to run it properly, and bam.
What was one of her early inspirations?
He had a telescope and he used to, take me up to the roof of his apartment and he would take images. I have a picture in my office of the moon that he took.
And to be seen, not just heard, her impressive sewing skills. They really are quite amazing.
Now, I also think it's a good skill to have if things go really awry in the world, right?
And her favourite food.
I mean, it's the perfect food, isn't it? When it comes to anything except health.
All in this episode of The Microscopists. Hi, welcome to this version of The Microscopists with Jennifer Waters over from the Nikon Imaging Center at the Harvard Medical School. So, Jennifer, how's you today?
I'm doing well. Thanks so much for having me.
I'm going to start off actually because you've actually recently-ish, just over a year ago, I think, got a Chang Zuckerberg award.
I did, yeah. Life-changing.
There's two things here because not everyone will be aware of the Chang Zuckerberg initiatives, and not everyone will be aware of what you've been funded to do. So could you explain a bit about the award, the Chang Zuckerberg initiative to start with?
Sure. So Chang Zuckerberg Initiative, as the name implies, is Priscilla Chang and Mark Zuckerberg's philanthropic effort. Oh, no, I'm doing it, Peter.
Keep going.
It's Priscilla Chang and Mark Zuckerberg's philanthropic effort. And so they're putting money into science and justice and education. And they are super psyched about imaging. I say often, I don't know what I did to deserve the fact that for whatever reason, they have decided that imaging is the most important thing, and they're putting tons of money into it. So it's fantastic. And not only that, but they recognized, and as far as I know, they're the first funding agency, certainly in the US, to recognize that positions like ours should be funded. And so yeah, the grant announcement came out and I read it and I thought, this is exactly what I do with my life. And it was the first time I had ever read a grant, call for proposals that sounded like me. So I wasn't sure if they were looking for like junior people. It wasn't super clear, or, old people like me, but I went for it. And I'm super glad I did because it's been fantastic. It's really enabled me to do some of the things that I've been wanting to do.
If you classify yourself as old, that makes me really old, and that's not a good thing to think about.
Well, I'm probably older than you think I am, Peter.
And I'm probably younger than I actually look.
Interesting. I'm tempted to compare numbers. I'm willing if you are.
We should go in the chat and just put them up in a minute.
Okay. I will tell you that I have a big birthday coming this year.
Oh, wow. So
a really big one.
I'm guessing that's not 40.
No, it's 30.
So, oh yeah, brilliant. You're older than me. Anyway.
No, I turned 50 in June.
You're a couple of years ahead of me.
Yeah.
Which is quite good, actually. We'll come back to that in a moment, actually, because there's some relevance to it. Actually, how our careers have been quite similar in their timings.
Yeah.
As well. But so you've got the Chang Zuckerberg Award. What is it to do?
It pays my salary for 5 years, and full salary. And there's travel money, and there is also they've given us sort of additional money we can apply for. So last year not last year. Last year we didn't do anything. Previously, they let us like apply for extra money to visit other imaging scientists in the program. So yeah, it's been great. When I applied for the grant, I told Harvard that if I get it, I'd like to hire someone additional in my group. So I was able to do that, which was awesome and has freed up my time, as I said, to do some of the little projects that I had been thinking about for a while.
And what sort of projects are they?
Well, Microcourses is one of them. So this is my YouTube channel. I make short videos on sort of fundamental concepts in microscopy. And it's something I've been wanting to do for a while. I've learned a lot about things on YouTube. And I sort of see it as a medium that is really accessible, and you know, I envision like the graduate students sitting in their microscope room waiting for their time lapse to finish and watching this, you know, 10-minute video on numerical aperture. So I feel like it's the kind of thing that is easily digestible.
Okay, so the importance of this, it's not obviously just for your students, your users of your microscope. So you run a facility which we'll come to. But this is obviously, and hence the award for the initiative, this has global appeal. And it's really educating about the fundamentals of microscopy and science in general, because microscopes are fundamentally vital and underpin so much science research across the disciplines, actually.
Yeah.
That now people can access this because it is on YouTube across the world. And I think that bit's really important. The biggest challenge, how have you managed to distribute, to get people to actually watch it? Because it's not just because you put it on YouTube, doesn't mean people will know it's there and useful. So how have you found that? Twitter.
I mean, Twitter has been amazing. I mean, I have to say, I'm not a huge social media fan. I don't really use it in a personal way, but it's been amazing for you know, getting the word out on these projects. So yeah, I think it's all Twitter.
Yeah, and I think we've had this conversation with other people in the past. Twitter isn't about, for a lot of scientists, it's one of the best places, one of the best resources for learning, finding out new job potentials, latest publications, courses, and access to materials such as your own. So it's not about a conscious of thought, which is where maybe it started with celebrities. It's very much a very useful tool.
It's super useful. I'm always telling people you really have to be on Twitter. I mean, I hear about so many things via Twitter.
So what's your Twitter tag? Go on, what's your tag for it so they can?
@GenC_Waters.
And at York Bioimaging on my side. They're fantastic resources. There's lots of fantastic out there. So quickly pick them up as it goes along. So you've done that, but actually, so your passion for microscopy is helped, and your passion for teaching. But that probably comes at the moment. Your current job is at Harvard, but running the imaging center there itself, and there's obviously a lot of teaching. So why are you running an imaging facility, an imaging center? How did you get there?
How did I wind up here? Okay, so let's go way back. Is that okay?
Yeah, go on.
Yeah, so I went to SUNY Albany for undergrad, and
what do you study?
I studied biology. And you know, honestly, like at the time, I had no plans, no big plans for the future. I grew up in a family. I was the first person in my family to go to college, so it was never something that was talked about in my home or expected. And to be honest, I just sort of, you know, I did well in school, and I just kind of followed a couple of friends who also went to college. And toward, I think it was my junior year, I heard that you could do something called undergraduate research. And, you know, it was explained to me what this is. You can get credit for doing research in somebody's lab. It sounded fantastic, right? So having never heard of it before. So I did that, and I wound up in Conley Reeder's lab, who is a microscopist, and did a lot of DIC imaging of cells in mitosis, using technology that no one ever uses anymore, except the DIC part. and took a lot of, 2-color fluorescence images on film, and loved it. I just loved it. I wound up working for Conley as a technician for a year after. And basically the people in his lab convinced me that I should go to graduate school. It was never something I imagined even when I went to college, but they convinced me I could do it, And so I looked around. I was in love with microscopes at that point. So I really wanted to wind up in a microscopy lab. And I applied to a few places specifically because there was a microscopist there I wanted to work with. And I wound up in Ted Salmon's lab at UNC Chapel Hill, go Tar Heels. And amazing experience. Ted, it is a biology program, was a biology program. I do have a degree in biology, but Ted's an engineer in training. And so he had a very technical approach to microscopy. We all knew our microscopes in and out. And Ted taught in a lot of microscopy courses. And I was the first one to volunteer to TA. I TA'd a lot for him. So when I was finishing towards the end of graduate school, I started to feel like I wasn't curious enough about any particular biological problem. Like I don't feel like I have that sort of intense curiosity that I think you need to really drive a research program. And so I started thinking, like, what do I want to do? And I knew I loved the microscopes. At the time, I didn't know anybody running a core facility that had a PhD. At the time, it was a technician, maybe a master's degree. So that didn't enter my brain. But basically what happened was one of the vendors who knew I was looking for a job learned that Wake Forest University, which is also in North Carolina, was looking for somebody to teach microscopy. So Nina Allen had just left there and left a sort of hole. She left a couple of microscopes behind. So I went there as an adjunct faculty member to teach a microscopy course. So that was the first time I developed and taught a microscopy course on my own. And I was there for 2.5, 3 years, I think. And then the job I have now opened up. And it was, I remember interviewing and asking every person I interviewed with, why aren't you hiring a technician? Because I wanted to make sure we're all on board. Like I could see why they should do this, right? And they had exactly the right answers. And so I felt really confident that it was going to be a challenging and interesting job. And so I took it.
So it's a big step, but actually it's very similar to myself. You started that in 2001.
2001, the core opened.
Yep.
Yeah, that's when I started my job. Yep.
And actually, I took over the run. It was a new whole infrastructure of core facilities, all as one unit being put together at York. And I was asked. I went for the job for the imaging and cytometry core there in 2002. And so only a year behind. And it was a very different initiative. But actually, I didn't ask that question. It was a very good question. But I kind of saw the potential that actually, if you're a microscopist, you've got to be very careful here. There's technicians, and it doesn't matter what technical post, there's all sorts of skill sets that everyone brings to it.
Absolutely, sure.
And I could see the potential of using the postdoctoral experience to drive research and enable research and bring that postdoctoral expertise to lots of different science. Instead of addressing one scientific question, it's addressing lots of scientific questions. And that's why they're such fantastic environments to work in. Mm-hmm. So have you had many difficulties in dealing with the variety of sample types, the variety of different research projects that have come through your doors?
Oh, sure. There are things that come in that are very challenging. And probably one of the most challenging things for me, really, in doing this job for so long, you never could have told me I'd be here in the same job 20 years later. But I still love it. And it's an incredible place to be. And so, for me, the hardest thing has actually been seeing how things have changed in microscopy over the years. And like when I first started, people would come in and everybody does a consultation initially with me and now with other people from my group as well as me. And, we talk about what they want to do, of course, their experiment. We think about how it might be done and controls and this and that. And it used to be that I could be like, oh, wait, we can do it way better than this. we're going to use a motorized stage and we got this and that, and they get all excited. And now they come in and they want to do, storm in a live spheroid, right? And so I find myself having to break a lot of hearts. And so it's gotten a little more like, you feel bad. But at the same time, what I tell myself is you are saving these people a hell of a lot of time, right? And effort by steering them in the right direction. So, so I think that's what I think of when I think of hard is the people that come in with an idea for an experiment they're really excited about, and I have to explain to them why it's probably not worth their time to even try. I'm always happy to test things with people. In fact, I like it when people are skeptical. And so I love a good skeptic. And so happy to test things, even if I think they're impossible. But yeah, those are the things that are hard.
You can be optimistic as well as skeptical at the same time.
Absolutely. Although no one's ever accused me of being optimistic. I consider myself a realist, but I think probably I'm unpessimistic.
But I'll say, it's very similar problems. Mm-hmm. So what do you find? What technique? What microscope technique is your favorite technique?
I have to say the appropriate one for the specimen.
Oh.
All right, that wasn't fair. I will give a real answer. Wide field. Wide field is an incredibly light efficient technique, right? You are getting as many photons out of the sample as most microscopes will allow, in a well set up wide field microscope. And the truth is it's the best choice for a lot of samples and a lot of experiments. It's going to get you, highest signal to noise ratio, for many applications, less photo bleaching because you need less illumination, et cetera. So I just still, I love wide field. The new techniques are exciting as well, of course, but I feel like wide field is overlooked too much.
I never had a Nobel Prize for it, I don't think.
Isn't that sad?
If you think about it. But then nor has gone both.
There's a lot of things in microscopy that it's sort of shocking that have not gotten Nobel Prizes that have had very, very high impact. Yeah,
absolutely. Now, I'll got to be careful how this looks, because I'll just ask you what your favorite technique is. What technique do you least like?
Oh,
that's a difficult one, isn't it?
I mean, there's no technique that I hate, things that are really prone to artifacts are I have the hardest time with. Not that those aren't things that can be worked around and paid attention to, but like for me, from my perspective, usability is so important. Right. And so when techniques come out and there's a lot in the super res space like this. Okay. When these techniques come out and they, the developer and the, down the line, the commercial companies that commercialize the instrument, they tell you all the great things about it. Right. But, there are always compromises, right? There's always things that you have to worry about and you have to test for. And so I think those techniques that from somebody who doesn't have a strong microscopy background, they look at it and think, I need to do that technique, but the limitations of it, the really like tons of limitations of it are not so apparent. So I'm not giving you an individual, single technique there, but I think those are the kinds of things that
less fond of. more skeptical of. Maybe that's a better word for it.
Yeah, I am. I'm skeptical
of the results.
I've never seen a microscopy image I wasn't skeptical of.
So, coming back to the start, what was your inspiration? Or who inspires you? Who inside and outside of work? Mm-hmm. Who are the characters? Who are the people? What inspires you?
Yeah. I think, this only occurred to me when I was about 30, to be honest. But when I was a kid, my dad was, as I said, he didn't go to college, but he was a big reader. He read a lot, and he was into science. We used to watch like Carl Sagan videos together and things like that. And I thought science was super interesting. And he had a telescope, and he used to take me up to the roof of his apartment, and he would take images. I have a picture in my office of the moon that he took. And there was a point where it clicked to me that, this must be in there somewhere, this idea of taking images through optics, right? So I feel certain that was an initial inspiration.
You do realize a microscope isn't a telescope, though.
I do. They're quite different, but they also have some similarities. So
a lot.
Yeah. And honestly, right now, at this point, I could maybe talk about other people throughout, but my group inspires me the most right now. I have the best group I've ever had right now, I would say. I've had a lot of great people come through, but.
So is this a lot of your group?
No, that is not my group. That is our last Cold Spring Harbor course. Actually, so some of my group is going to be in there, maybe everybody.
Well, actually, so for your...
Let me put my glasses on.
I can go to your group, I can come back. So this must be some of your lab.
Well, yes. So that is, we've got Tally Lambert there, who I think many of you know, and Anna Jost in the middle. And then not part of my group, but Hunter Elliott there on my right.
And what are they to these? I've got to say, they look like they're in a uniform of yellow.
They are in a costume. So if anybody's looked around our website, you might know that our microscopes are named after characters from the show Arrested Development. And anybody who as a fan of the show will look at our website and sort of know that we work hard to put the appropriate character with the appropriate microscope. But this was one of our departmental retreats, and we had it was around Halloween, and we had a costume contest. So we were dressed up as the workers in the banana stand, which again, if you're a fan of Arrested Development, which many people are, will know exactly what I mean, and you'll think it's super cool.
So I noticed you took the picture. I take it you were also in the costume?
Honestly, I wasn't. I would have been, but sadly I hurt my back, and I wound up not being able to go. But yeah.
So thinking about when you can't get into work, I presume this is probably from a lockdown period, and
this is all of us. This is the current group, plus not all of our pets, but some of them. So yeah, me of course, and then. Sorry?
Who have we got in the picture? Give them a shout out, because obviously these are the people that really do the work now, because obviously you're just making YouTubes all the time. So
yeah, I don't do much of anything, to be quite honest, anymore. These people do it all, right? Yeah, so there's me up there with my cats, and Federico Gasparoli. So he's our newest fellow. He has started since the pandemic. He's Italian. He came from Scotland. And he had to wait a very long time, poor guy, for his visa. He's yet to know what the core is like without, a mask and 6 feet away from everybody. But great guy, super enthusiastic. He fit right in right away. So Anna Jost is holding the adorable tuxedo cat up in the corner there. And Anna was, that's her. Anna was the second fellow in the Advanced Microscopy Fellowship program that I run. And actually, one of the reasons why I say the CZI award was life-changing is because what I did was hire Anna. And so her official title is Associate Director of Imaging Education. And so she's also very passionate about teaching like I am. And it's been awesome to have somebody who likes to think and talk about teaching approaches as much as I do. So that's Anna. And then this way here. Oh, yep, that's Tally Lambert again with 2 out of his 3 cats. Yep. And on the other, and yeah, so Tally, I could talk a little bit more about Tally. Tally has a bit of a history. So he was actually a student in my first Cold Spring Harbor course in 2011. And anybody who knows Tally knows that he is incredibly smart. So he really stood out as a student, and we invited him back as a TA for the next course. And you know, of course, he was once again fantastic, and he wound up the first of the fellows in my program. So we were also very lucky to be able to recruit and hire him, and he runs our most advanced microscopes. He works with our users who are, you know, sort of doing the most complicated experiments. And, you know, he's behind FP Base and is a major contributor to Nipari now. Yeah, great guy. And then last we have our second fellow, Riley Walsh. Riley, also awesome. She's very interested in teaching. She's obsessed with sim. And yeah, they're all just fantastic. And like I say, they're super inspiring, particularly the fellows. They're so excited. You know, they just want to do everything. And they're just such a pleasure to have around.
You got the fellows, and you mentioned on the cold, just for clarity, you got the TAs, which I presume are teaching assistants.
Yes, teaching assistants.
Yeah. And these are, I think, a close-up of your cats.
Yeah, those are my fur babies. So I am a proud crazy cat lady, and that's Chloe and Olivia. So I adopted them almost 4 years ago now from a local shelter, and they were you know, street cats. So we had a little bit of a breaking in period, but they're absolutely adorable. And have been a great comfort to me in the last year as I've been stuck at home primarily.
And I have there was a picture that has not come out, which is drat. So I was going to say, because I also have a picture of you with the cats actually on a sewing machine, and the picture of the sewing machine isn't there for some reason. But this is either a deconstructed pair of jeans on your floor, or you have cut the material out and started to sew your own jeans.
Yeah. So one of my passions outside of microscopy is sewing. I absolutely love to sew. I've made the majority of my wardrobe.
There you go.
I am. Yep, that's me sewing at my Bernina sewing machine. I actually have 4 sewing machines. That one was my first one and the one I use the most.
So why do you need the other 3?
So I have 2 standard sewing machines. So when you make something like jeans, the last picture, which we can maybe get back to, but you are using 2 different types of thread. You're using the thread that you use for construction, and then you're using the sort of the thread that shows on the outside of your jeans, right? The, you know, usually yellow or coppery or color. And so it's just faster and easier to sort of go back and forth between the 2 machines rather than rethread each time. So that's a little bit of a luxury. And then I have what's called a serger, which finishes the edges of fabric so they don't fray. And they're also used to construct knit garments like t-shirts and things that are stretchy. And then I have what's called a cover stitch machine, which is for hemming garments primarily. That's what I use it for.
So it's just like microscopes, a different microscope for different experiments. You now have a different sewing machine for each purpose of sewing.
Exactly. So, they each have their own purpose. Yes.
So the shirt you're wearing today, homemade or purchased?
Yes. Nope, homemade.
No.
Yep. Yeah, and the picture behind you is a pair of jeans that I'm in the middle of constructing. So I've done the fly and the pockets and the back pockets and the inseam, and the next step is to, sew the seams down the side and hem them and put the waistband and all that on it. So it's so fun. And it's one of those things that my brain is, like many of us, I have a hard time turning it off. It's just cranking all the time, right? And so it's something that I can do that requires a lot of attention, or you screw up, which is frustrating. You have to backtrack. And so I love it because just turn off, I just engross myself in, constructing this garment, and it's super fun. And then I have like clothes that fit me.
Either that or Harvard aren't paying you enough. So
Well,
actually,
I also think it's a good skill to have if like, things go really awry in the world, right?
I think I'd rather farm, maybe, if things go really wrong. I think about the food.
We'll need farmers too, but we need people to make our clothes.
So if you're listening to this podcast, do go and watch it on the YouTube channel, because the shirt that Jennifer's wearing right now is awesome. I'm very impressed. It's a very different skill.
It's really fun. I mean, there's different aspects of it too that are fun, like the construction, like, when there's hardware like this, there's hammers involved. I mean, it's fun.
There's so many parallels to the science world.
Yeah.
All the instruments, all the tools, even hammers and your reagents that you're buying in.
Exactly. It does feel like a scientific process. There's problem solving, there's planning, there's finding the right components, and of course the machines, which of course I'm always calibrating and cleaning and making sure they're working optimally.
So what else actually, you've got a ton of plants. I was going to say, what else do you like when you're outside of work? But you've got a ton of plants behind you. And actually, I think you sent me some pictures of lots of plants.
That's one of my windows. Yeah, I love houseplants. I have a lot of them. I have over 100. There's a couple of like genera that I like to collect. And so it's a little bit of an addiction, but it's another hobby, I'm a hobbyist.
What genera of plants do you?
I love Peperomia in particular. They're my favorite. The image you just showed is my Hoya window. I like Hoyas a lot too. It's sort of like their growth pattern. This if you're looking at me, the 3 on my that you can see on my shelf here are Peperomia. So, and I have some ficus over here. Again, it's one of those things that is just
the biologist.
It's a little bit of the biology for sure, and it's also it's super relaxing. My mornings, I live alone, and my mornings are get up, make my coffee, wander around the house and check out the new leaves and water, and I find the whole process very sort of zen, which is something that I'm a little bit intense, so zen is good for me.
So I noticed this picture, which you have of more plants, but I also noticed, I presume in your house somewhere. I presume this is your house. I noticed a picture hanging on the wall. Yeah. So actually, I had to zoom in on this. So I kind of edited your picture a bit and zoomed in.
Yeah. So another hobby, which honestly has fallen a little bit on the wayside, is painting.
That's a painting.
Yeah, I painted that.
That's awesome.
Yeah.
Okay, so explain. It looks really great. Like microscopists, I've got a pretty good idea what it is, but go and describe this picture for those actually listening.
Thank you. Thanks. I mean, I'm certainly no professional artist, but it is fun. I mean, in general, I like to make things, so it's something I haven't been doing too much of lately, but I actually have an idea for a painting that's been brewing. But yeah, these are point spread functions. I call this one pleasure and pain. You've got a sort of artist rendition, of course, of a perfect point spread function, and then a highly spherically aberrated point spread function.
Yeah, I prefer the perfect one. It looks like I'm radiant. It looks like I've got light shining from my head. Quite good.
Don't we all? I have a little bit of I don't know, point spread functions. Don't we all love them? And I did a little bit of a point spread function series. I have a point spread function tattoo. I think some people know that, but on my back as well.
I won't ask you to show us right now.
The students at QI get to see it.
Right, I wanted Jennifer Waters' picture in my house now. That's something that I've got to get. It's a beautiful picture. It captures it really nicely.
Thank you.
At least your aberrated one wasn't as bad as it could have been after a bad user.
Yeah, can definitely get worse, can't it?
So back into work. Have you ever, what has been the most difficult time you've encountered at work, and how have you overcome that?
That's a good question. I think, for me, probably the hardest times that I've had have been learning how to manage people. That was, I really lucked out when I first started my job, because the first person I hired, who I have to mention, is a woman, Laura Petrak. And I hired as a technician. She worked for me for 11 years. She worked her way up. And she was absolutely fantastic. And we worked very well together. She now works for Oka Lab. And she kind of made me think it was easy, and I've had fantastic people throughout my career working with me, but it's managing people can be difficult. And that was learning that process and learning how to do that well was hard. That was hard.
Still learning?
Me. Absolutely. Yes. I do think I've gotten better at it. But yeah, and honestly, my current group makes it easy because they all have, they're just such hard workers and very, very creative and enthusiastic. So and hard on themselves. I'm quite hard on myself, and these guys are too. So most of what I'm doing is really just telling them they're doing a great job.
So
they make it easy on me.
How did you learn the skills of managing staff?
I actually took classes at one point because it was becoming clear to me that I didn't, basically what's happened is when you have somebody that works differently than you do, it's trying to understand that and get the best, get the best relationship one can given that. And I took classes. It turns out this is a field that people study, right? And I learned quite a bit from that. Simple things like have a one-on-one meeting with each person every week. And I started every week. I have a one-on-one meeting with each person from my group. I mean, there's only 5 of us, so it's not hard, but it is really has been really good, I think. Sometimes the meetings, I always tell everybody they're completely optional. If there's nothing you want to chat about, you can just not come. And sometimes they're 20 minutes, and sometimes they're an hour and a half. It's just a time that put aside every week that we can touch base. And it's actually been really important now, right, when we don't see each other every day, unfortunately.
I think my team would hate me if I did that. I think they have enough of my voice just on the lab meetings themselves, which is a couple of times a week at the moment.
Yeah. Well, it tends to be pretty informal, and some of it's just chit chat, and but yeah, it's really I don't mean it. I absolutely hate micromanaging. It's I despise it. Right. I work best.
I get it. Don't worry. I do get it.
Yes. Yeah. But no, what I want to say is I really like people who are just sort of doing their own thing. Right. So it's nice to have a place to come together and talk about what they're doing, so we just sort of chit chat, and yeah, I like it. I think they do too, but.
So difficulties at work, managing, throughout your career, what has been the biggest highlight, the most enjoyable period that you've had to date?
Starting the Cold Spring Harbor course was, that was a really big deal to me. So I had taught, as I mentioned, I was a teaching assistant for Ted Salmon for many courses when I was a graduate student. And when I got the job at Harvard, I thought, oh geez, I'm at Harvard now, I better, brush up on stuff. And so I contacted the directors of the course that I had taken as a graduate student, which is at the MBL, the AQM course, and asked if I could come and just kind of hang out. And I wound up going to that course for 9 years. And at the same time, of course, I was developing curriculum at Harvard, and I was by the end teaching a graduate course in microscopy at Harvard. And the directors of that course directed it for a very long time, and so I just got to the point I wanted to do my own thing. I had a lot of ideas, and I just, honestly just was getting frustrated kind of with somebody else's vision. I had my own, and so I decided to leave. And I should say I was teaching in other microscopy courses as well, a couple of different courses at Cold Spring Harbor, one by John Murray, for example. And at the time, at Cold Spring Harbor, because I was a core facility person and didn't have the word professor in my title, I was a teaching assistant in the course, even though I was acting as an instructor, right? So at the time, people in core facilities running a course like this was not a thing. And so I left.
Yeah, you put that down to, I guess, just literally name snobbery.
Absolutely. Yeah, I think
the title, not of the capabilities at that point.
Yeah, I mean, and yeah, it's just a perception, right? Of this is the realm of the professors, the teaching of the grad students and professors, right? You core facility people are there to help know how to use the microscope, work, train them how to use the microscope. But I think that was more the perception at the time. Obviously, it's changed dramatically in the years since, but I knew I could run this course. That's sort of how I felt. I knew I could do it. I wanted to do it, and I actually, I felt like I was really good at it. And I, as a core facility person, you're putting more time into learning how to teach this stuff, right? So I was going to these courses, and to be quite honest, there were people giving really bad lectures, like excellent microscopists, name recognition, but terrible lectures. And for me, I was like, I just want a course where every lecture is really good, and the students learn everything, and nobody makes any assumptions about what they already know going into it. I had a vision. So I left, I decided to just leave the course. I didn't want to do it anymore after the 9 years. It was long enough. And 2 months later, Cold Spring Harbor called me and asked me if I wanted to start a course. And I was over the moon, because I don't know that there was anybody else running a course, one of these courses that was running a core facility. I don't think any microscopists, of course, since then there are, but so I was really proud. It was something I felt like I worked hard to do, and that was a big moment. And I'm also very proud of the Advanced Microscopy Fellowship as well. I think that's been really successful. So
and just pointing out, and you've obviously got 2 of those fellows now working with you. Mm-hmm. And the courses aren't just about teaching people how to use microscopes. They also motivate and show people that there's a career in microscopy as well, and it can really change people's careers, as it has through your career, and actually a couple of your staff. They have had their futures inspired. And I'm sure there's many others that have gone on the courses and now similarly been inspired to become microscopists, not biologists or life scientists using a microscope, but actually becoming a microscopist using biology for their microscopy.
It's so satisfying. It's so satisfying to, I love teaching at Harvard too, but it's smaller scale. We mostly teach 1-day workshops. And so Cold Spring Harbor, and particularly short, intense courses, are so satisfying to see how much they learn over the course of the 2 weeks. And they're all, many of them are already doing a microscopy experiment. And some of them leave, this was actually Tally, left realizing he was doing a lot of things wrong just because he didn't know, the details of the equipment. And so it's really satisfying for them to know they're going to go home and, do a better job. And we definitely have had alumni who have gone on to, be in core facilities that weren't planning on it. So yeah, it's fun.
And you mentioned how, going back a little bit and just bring this up now, how perceptions of people who work in facilities under the banner of technicians, I will really annoy, I know, one of our friends by saying we offer a service in some respects, and we are there to serve in some respects. But serving and collaborating can be the same thing, I think, and it's teamwork, and its terminologies can really upset some people. And I think we're in a much more open world now, but you must have faced a lot of, positional discrimination.
Yeah,
Does
absolutely.
that make sense? Your pseudo status, even though salary-wise, you'll be on quite a senior equivalent salary. I'm not going to go there, which is equivalent to some of the very senior researchers, lecturers, so forth. But because the title, you said, is because you haven't got the title professor.
Right.
There's sometimes a perception. I think that is less so now.
Mm-hmm.
How have you found that over in the States?
It's changed a lot. I mean, part of the reason why, as I said, I asked everybody at Harvard, at Harvard Medical School when I interviewed, why not hire a technician? And it was the fact that they understood the additional, knowledge that you can bring to helping people on the microscope if you have the experience of having done research yourself. And so, but nonetheless, I was a core facility director, and that was quite different. And, I have memories of won't name names, but of a very well-known senior faculty member saying to me early on, like, I don't know, I wouldn't want to do experiments and having my people do experiments in the core if I don't understand those experiments. And I feel like that's changed. I feel like there's a lot more trust. I'm sure ultimately they understand. I don't mean to make it sound like, but I think the faculty are much more, and it's particularly junior faculty, right? It's definitely junior faculty come in and treat us like colleagues immediately, right? So that has changed, I think, a lot with the younger generations. But one of the reasons why I've stayed at Harvard this whole time, the reason why I haven't left, is because I do feel like a colleague there. Over the years, it has become where, there are labs at Harvard Medical School who do all of their imaging in the core facility, who don't have microscopes in their lab, who rely on, my group's expertise to guide their trainees. And so I don't feel that way locally. I don't feel that way at all locally, or very, very rarely. I feel like a colleague. And I get, I don't mind saying that it's a service. Like, we are here to help you, that's fine. But what I do care is that I get the respect that I deserve based on my expertise and experience. And that I feel for sure here.
And you touched on that, going back a bit. There'll be a lot of postdocs in the world that will think, oh, well, why would you want to give up research? Wait for it. Why would you give up research to just use a technology? And this could be any technology. It could be a genomics platform, a mass spectrometer. It could be all sorts of different equipment types. What would you say? Would you not want to have a project and make that your, yes, you help others, but would you not like a question that you're resolving yourself? What would be your answer to that?
Not a biological question, to be honest. There hasn't been a biological question come across that I've gotten. I certainly feel the excitement when a user is excited about their data, and but for me, I'm much more interested in understanding how microscopes. I'm much more interested in deeply understanding them and testing the things that are not clear, right? And so, that's what interests me more. And I think as much, I think that's much more useful, better use of my time. So, yeah.
Yeah. So my counter to the argument is, well, actually, why would you want to only solve one biological question when you can help solve lots of biological questions?
Yeah, exactly.
And you mentioned it, the excitement when a user, and yourself, because you've helped them, sees something for the first time, and it answers a question they've had for ages about the fundamentals of life, and you've seen one of those questions answered in front of your eyes.
Yeah.
And maybe you don't appreciate it to the same depth as a user, but you know what they were looking for because you talked to them, obviously. And when they see it and you see it, or you point it out, say, no, I can see it. The look of excitement on their eyes,
It's infectious for sure.
Accumulation, but it is exciting for us too. Yeah, because we're part of that team. We are part of so many teams.
Yes. Yeah. Yeah, it's true. I mean, I think one of the things that, I mentioned I'm a skeptic. I think that's one of the things that my group and I sort of share. We're all quite skeptical. And I think that actually matches very well with the researcher, right? Because they're so excited and they're so trying to get the answer, and we can be there gently reminding them, well, we still need to do that control, like, let's double check this and that. So it does very much feel like a collaboration as you're working together. And one of the things I also get really excited about is when we have somebody come to the core who has been trying to get an experiment to work, like in their lab, and have really been struggling, and we can help them. We can turn that around and put them on the right microscope and show them how to run it properly, and bam, it's, the signal they didn't think existed is there. So that's super satisfying too.
How often do you get on the microscope still?
Oh, not that much. Not that much, I'm afraid. Yeah.
Do you miss it?
I do at times. What I really miss is feeling intimately involved with every single microscope in my core. I mean, for most of the time I've been here, I was the one who knew the most about every microscope, like all the intricacies of how it's set up and such. And that's not the case anymore, So that's kind of, thankfully, I have people that I very much trust who hold that information. But, I'm at a point now where I really like to talk about imaging, and I really like to talk about, experimental design and approaches. And obviously I love to teach. And so while there are times for sure that I miss it, I like the things that I'm focusing on right now. And I live through the fellows who are so excited about all the time they get to spend on our microscopes.
So you don't get much time on the microscope. You've got a great team. You've got the Chang Zuckerberg Initiative Award to setting up those. What does the future hold? Where do you see yourself going? I'll start there. You're 49 years old. You're coming on 50. You confessed to that already. You're midlife crisis time. What are you going to do? Yeah. What's your next challenge?
My next challenge. So, I do want. I tell myself the microcourses are my book. I want to cover all the topics. I've been really slacking off in this pandemic. It's been hard, isn't it? So, but I have a lot of topics that I want to cover. So that is, to me, a big project that I'll be working on for years, I think. I have been talking with, I'm not quite sure. I always have like ideas brewing, and I'm not sure which direction I'm going to go in, but I have been talking with 3 of my imaging scientist colleagues, Abhishek Kumar, Martin Fisher, and Aaron Taylor, about sort of putting together there's so much so many, especially now with COVID, lectures and learning materials for microscopy online, right? There's tons of them. And I'd like to put together a website that's sort of curated by, people who do microscopy teaching, set of lectures. And I'm also really, and what I mean by that is we would sort of go through what's out there and pick the ones that we like the best and think cover the kinds of material we want to be part of this curriculum, and put that together. And yeah, there's a lot more to that idea, but the other thing I want to do, and somewhat related to this, is I want to start I want to help junior imaging scientists, junior core directors who are starting up teaching. I want to help share curriculum and such with them. So I have an idea. I actually have a website I'm working on right now that I'm gonna share the Microcourses slides. So that's almost up and running.
I have to ask, the YouTube channel for this is called
Microcourses. Microcourses.
Just to make sure we're clear, and that's not a mu, but that is an M-O-M-I-C-R.
That is M-I-C-R-O courses, all one word. Yep.
Right. So we are unbelievably coming up to the hour.
Wow.
I did say it'd go fast. I've got some quick questions for you though. What is your favorite food?
My favorite food. I think I have to say pepperoni pizza, as much as I wish it wasn't.
Ah, absolutely brilliant. Yeah, I'm with you all the way.
I mean, it's it's the perfect food, isn't it? When it comes to anything except health.
No, do you know what? It's the perfect fuel for drinking on an evening or just long running on a Saturday. It's full of carbs, sugar, salt, fats. So
Yeah.
perfect. Yeah, I love it. Every
Yep.
Friday night, it's a religion.
Is that? Yeah, actually, when I have pizza, it's a Friday night thing. So
Yeah, since undergraduate days, that's the one thing I will not allow to change. So, what is your what food do you most dislike? Imagine you're taking out on a course or a meal.
yeah,
Nightmare dish to have served to you.
I am not adventurous when it comes to seafood. I did not grow up eating seafood, and so yeah, there's quite a few things that people love that kind of. I will try anything, but there are plenty of things in that genre that I don't go back to.
Yeah, I again, I would agree with that. I remember one Elmy when it was a seafood restaurant and it was crabs, lobster, and shells, and that was it. That was it. There was
Yeah,
nothing else that night, and one slice of bread. That was a
yeah,
hungry evening that night.
right. Yeah, like I can get by, but it's never satisfying.
But look on the bright side, there's always drink wherever you go. So what's your favorite drink?
It's true. I like cocktails. So let's see. Martinez, I think, is probably my favorite.
Okay.
Also like a good Negroni. Yeah.
And least favorite drink?
Bartles and James wine coolers. Do they still make those?
What was that drink?
When I was in, oh God, it was high school, college, there were these Bartles and James wine coolers. They were, bottled like wine and juice or something.
Okay.
Things of that genre I do not like.
I like the fact that your favorite and least favorite are both alcoholic, but okay. You were straight there with alcohol. Forget the coffee, the teas, the hot chocolate.
Oh yeah, well, coffee. I mean, isn't that a given? Is coffee like? I feel like that's a requirement.
Okay, and what about so you've got your food, you sit down, you've got your drink.
Mm-hmm.
So what would you want? What movie would you watch? What would be your favorite? What's your favorite movie of all time?
I don't know if I have a favorite movie of all time. I love documentaries. That's usually what I'm watching.
Okay.
And I like this sort of documentary series that there's more and more of now. So that's usually, yeah. I do watch movies. I've watched a couple of good ones lately, but documentaries are the thing that I.
And what content type though? I mean, there's documentaries about all sorts of things. What
anything. If it's a good documentary, I like it. Anything. One of my favorite documentaries actually is King of Kong. Strongly recommended if you haven't seen it. It's about the sort of world champion Donkey Kong players. And it's just a really, what I think is the best is when it's a topic you wouldn't think you were interested in, because I don't care about, competitive video gaming. But, when they can make it like fascinating and you go into this whole other world that you're not, part of, it's, yeah.
So you really did mean anything, didn't you, at that point?
Yes.
Okay, so you've watched a movie, you go to bed, you put some music on. What music would you listen to? Classical, rock, pop, indie? What would be your genre there?
Oh, wow. I honestly don't listen to a ton of music. If I do turn on music, it's going to be something like Norah Jones or Taylor Swift. It's like those sort of maybe girly music. I listen to a lot of podcasts, actually. That's usually if I've got my headphones in, water and plants, I've usually got a podcast on.
Okay. And then it's time to make yourself sleepy. So you pick up a book, or do you read books?
I do, not a ton, but I do. I used to be a really heavy reader, and yeah, but I do.
Okay. And finally, you're then going to wake up the next morning and you're going to get some clothes on.
Yes.
I think I know what the answer to this is going to be. What is your favorite item of clothing?
Well, yeah, it's definitely gonna be something I made, of course. And it changes a lot because I make clothes a lot. I mean, I have way more clothes than I possibly need, and I give them away a lot because I make way too many. Um, I think jeans really are my favorite thing to make. And so probably, I've probably made 15 pairs at this point, and but there's still a couple that they're starting to wear out. I love them so much. So probably, and I live in jeans, so
Denim. I did think that would be the answer.
yeah.
What's your least favorite item of clothing? Which you obviously don't own, because why would you own something you dislike that much?
Yeah, I actually did make a blazer one time just because I thought it would be a good thing to have. I'm not really a blazer person, and I also wanted to make one because I didn't know how to, so it's kind of challenging. But yeah, anything like dress. I'm just not a person who likes to dress up. Anything dressy. I like to wear a dress, like a casual dress in the summer, but I don't. Oh God, high heels. No, done.
So to end on, I usually ask if you've got a favorite joke. So actually, I have noticed that my shirt's actually quite cool. Bordering on moire fringes, I think. And I've heard a moire fringe song. So I'll give you a choice. Favorite joke or your moire fringe song, which I don't know. So
Peter. There are things that I'm good at and there are things that I'm not. And singing is way up there on the list. Tally has a great singing voice. So if he was, available, I would have him sing it for you. But it is a song, and Tally does sing it when he does his sim lecture. And I will tell you the lyrics, which is, when the moon hits your eyes like a big pizza pie, that's amore.
Oh, now I know what the song is as well. Oh, as in I know how it's gonna go.
Yes. Now it's in your head. You're gonna be listening to it all day.
Oh no. I am. I'm going to be running for hours tomorrow morning, and all I'm going to have is that in my head. Yeah, thanks, Jennifer. And on that note, Jennifer, thank you very much for joining me today. It's been great to catch up and have a chat with you, and I hope everyone's enjoyed listening or watching. Please don't forget to subscribe to the various channels. And I say this one's well worth watching. The pictures, the point spread function picture, so artistic. It was so brilliant, and
thank you so much.
It's brilliant. So, Jennifer, thank you very much.
Thank you, Peter. It was a lot of fun.
Thank you for listening to The Microscopists, a bite-sized bio podcast sponsored by Zeiss Microscopy. To view all audio and video recordings from this series, please visit bitesizebio.com/the-microscopists.